Samplex Lord Presence, sample exams on Biochemistry. Click the Start Quiz button to proceed.
Always do it on my own, so I gotta get through it. And the only thing I know is to love what I'm doing. Never give up, never slow, till I finally prove it. Never listen to the no's, I just wanna keep moving. Keep my head up when I act, head up, that's a fact. Never looking back, I'ma keep myself on track. Keep my head up, staying strong. Always
Quiz instructions. Before attempting, carefully read the question text. Then, choose the correct answer. Click on Submit to confirm your answer. Also, kindly subscribe to my channel and hit the like button. Comment for additional information on the questions and answers presented. Thank you. The breakdown of one molecule of a C16 fully saturated fatty acid, palmitic acid, by beta oxidation lead to the formation of 8 fat H2, 8 NADH, and 8 acetyl-CoA molecules, or 7 fat H2, 7 NADH, and 7 acetyl-CoA molecules, or 8 fat H2, 8 NADH, and 7 acetyl-CoA molecules, or 7 fat H2, 8 NADH, and 8 acetyl-CoA molecules, or 7 fat H2, 7 NADH, and 8 acetyl-CoA molecules. Correct. Well done. The breakdown of palmitic acid, C16, requires 7 cycles of beta oxidation, each producing 1 fat H2 and 1 NADH molecule, and results in the formation of 8 2-carbon acetyl-CoA molecules. What 20-carbon, polyunsaturated, omega-6 fatty acid serves as the precursor for the synthesis of the predominate series of prostaglandins, thromboxanes, and leukotrienes, collectively known as the eicosanoids, as shown? Is it linolenic acid, or arachidonic acid, or linoleic acid, or palmitic acid, or all of the above? Correct. Well done. The eicosanoic fatty acid, arachidonic acid, is the precursor of the predominant series of eicosanoids. Eicosanoids differ from endocrine hormones in that eicosanoids are produced in very small amounts in virtually all tissues, are not stored, act locally, and have their biologic actions mediated by cell membrane GPCR and changes in the concentration of cyclic AMP. Source: Lipincott's Illustrated Reviews Flashcards Biochemistry. All of the following statements about acetyl-CoA carboxylase are correct, except, does it catalyzes the rate-limiting step of fatty acid synthesis, or does it require biotin, or is it inhibited by cyclic AMP-mediated phosphorylation, or is it activated by palmityl-CoA, or does its content in a cell responds to changes in fat content in the diet? Correct. Well done. Acetyl-CoA carboxylase is activated by citrate and inhibited by long-chain fatty acyl-CoAs. Since cyclic AMP increases at times when energy is needed, it is consistent that a process that uses energy would be inhibited. Long-term control is related to enzyme synthesis and responds appropriately to dietary changes. Source: Devlin's Biochemistry with Clinical Correlation, 7th edition, pages 704 to 705. Which one of the following is a chain-breaking antioxidant? Is it glutathione peroxidase, or selenium, or superoxide dismutase, or EDTA, or catalase? Correct. Well done. Antioxidants fall into 2 classes: 1. Preventive antioxidants, which reduce the rate of chain initiation, and 2. Chain-breaking antioxidants, which interfere with chain propagation. Preventive antioxidants include catalase and other peroxidases such as glutathione peroxidase that react with ROOH, selenium, which is an essential component of glutathione peroxidase and regulates its activity, and chelators of metal ions such as ethylene acetate, EDTA, and ethylene pentocetate, DTPA. In vivo, the principal chain-breaking antioxidants are superoxide dismutase, which acts in the aqueous phase to trap superoxide-free radicals urate, and vitamin E, which acts in the lipid phase to trap ROO radicals. Source: Harper's Illustrated Biochemistry, 31st edition. Match these major apolipoproteins to their functions. Apolipoproteins A1, B100, C2, E. Functions are: it activates LCAT, binds to LDL receptor, cofactor for lipoprotein lipase, and mediates extra, remnant, uptake. Correct. Well done. Match these types of familial dyslipidemias to their disease descriptions: type 1, type 2A, 2B, type 3, type 4 and type 5. Disease descriptions: hyperchylomicronemia, hypercholesterolemia, combined hyperlipidemia, dysbeta-lipoproteinemia, hypertriglyceridemia, and mixed hypertriglyceridemia. Correct. Well done. A 40-year-old woman presents with an LDL serum level of 400, normal value less than 130, and a triglyceride level of 170, recommended level is less than 150. She is diagnosed with type 2 familial hypercholesterolemia. In this disorder, a mutated LDL receptor is formed, such that it cannot bind to LDL. Which of the following would the result be? Cellular HMG-CoA reductase activity is not inhibited, or the triglycerides in chylomicrons cannot be degraded, or the VLDL level in serum increases, or the HDL level in the serum increases. Correct. Well done. A patient with familial hypercholesterolemia, type 2A, is described to have a defective LDL receptors or mutation in ligand region of ApoB100. This manifests as an elevated LDL and cholesterol in the blood. This disease can result in atherosclerosis and coronary disease. From which metabolic intermediate does the synthesis of cholesterol begin with? Is it with acetyl-CoA, or HMG-CoA, or mevalonate, or squalene or laanosterol? Correct. Well done. Cholesterol synthesis starts with joining of two molecules of acetyl-CoA. These is to form acetoacetyl-CoA with the enzyme acetoacetyl-CoA thiolase eventually forming HMG-CoA and so on. All the others are intermediates further down the pathway. Source: Harper's Illustrated Biochemistry, 31st edition. Which of the following inborn error of lipid metabolism, sphingolipidases, is characterized by accumulation of GM2 ganglioside? Is it Niemann-Pick disease? Is it Tay-Sachs disease? Is it Gaucher's disease? Is it Fabry's disease? Correct. Well done. Examples of sphingolipidases. Refer to the image. The sphingolipidases, lipid storage diseases, are a group of inherited diseases that are caused by a genetic defect in the catabolism of lipids containing sphingosine. They are part of a larger group of lysosomal disorders and exhibit several constant features. 1. Complex lipids containing ceramide accumulate in cells, particularly neurons, causing neurodegeneration and shortening the lifespan. 2. The rate of synthesis of the stored lipid is normal. 3. The enzymatic defect is in the lysosomal degradation pathway of sphingolipids. 4. The extent to which the activity of the affected enzyme is decreased is similar in all tissues. There is no effective treatment for many of the diseases, although some success has been achieved with enzyme replacement therapy and bone marrow transplantation in the treatment of Gaucher and Fabry diseases. Source: Harper's Illustrated Biochemistry, 31st edition, pages 234 to 235.
No fake love, I want the real stuff. Everybody listen up, cause I'll only say it once. I'm gonna show you all the path. If you want it bad, I'm gonna show you where it's at. Yeah, how you can get it back.
This apolipoprotein is involved in the clearance of triacylglycerides from chylomicrons and VLDL by the enzyme lipoprotein lipase. Is it ApoE, ApoC2, ApoA1, or ApoB48? Correct. Well done. Beshiwap, you passed. Congratulations.
I'm sick of being screwed, feel my own adrenaline.